All right, we're back again, this time to move up a level in scale and describe the mechanics of the whole heart, or at least a superficial introductory view of the biomechanics of the whole heart. It's always fun to think about structure function, and we'll certainly do that. And it's also fun to think about the kind of behaviors that we have to understand and have to deal with. And one of them is of course the motion of the heart itself. And so it can be very helpful to see a picture of the heart in action. And so what we're going to see here is the picture of a heart contracting, and it will, it's an MRI slice. So we're looking from different sides. We'll have lateral views, so sagittal slices, as well as anterior views, or the classic 4-chamber view. And what you see here is obviously the torso, the entire torso, and you can see this is mostly a left ventricular view. You can see the dark, thick walls, the white on the exterior is fat, the light colored material going through there is the blood, and you can actually see the valve motion. Here's a classic 4-chamber view, in which you can see both valves, both the atrioventricular valves, so the tricuspid and the mitral valve, both in action. You can see the blood flowing briefly from the atria into the ventricles, and then this much slower contraction, bigger contraction in terms of volume of the ventricles. And you see how both ventricles contract at the same time. So this is really a nice image of the mechanics of the whole heart. And again, we can show other views, right ventricular views, and again, see this sort of contraction. We see how the heart really is anchored more or less at the apex and gets pulled down. Instead of the apex coming up, as you might naturally guess would be the kind of behavior that we have. But instead, it's really a pulling down of the base of the heart that allows the heart to shorten its long axis, we call it, but also contract around its circumference. So the volume gets noticeably smaller. The volume doesn't go to zero. I think that's obvious from these images too. And this is why the ejection fraction, the amount of blood that leaves the heart with every beat, doesn't get close to 100%. It's much lower than that, and it will also vary as a function of exercise, for example. But we consider 30 to be an important cutoff. Below that, that's abnormal function. So at rest, we'd be expecting to be sort of 45 to 50%, somewhat higher in exercise. So this, like I say, this is the behavior that we can see here that we really have to be able to explain. And like I say, it's always fun to think, what is it we have to explain? Now, I also want to use this particular case to describe the behavior, the differences with some other mammal, or other animal, not even just mammal, hearts that we also have to explain, right? If we're going to understand this system, we should be able to explain why it works differently in different animals. This is a deep well. I'll try and stay somewhat close to the surface so we don't get overwhelmed. But this chart here is kind of fun, I think, to look at, because it starts with the cow, and you've seen a cow heart, so you have some sense of what that involves, and then a human heart, and then a dog heart, a rabbit heart, and a rat heart. So this spans the spectrum of all mammals in this case. And we see the weight going from something in the range of 400 kilograms down to less than half a kilogram, around half a kilogram, and that's a decrease as we go from the larger species to the smaller species, no surprise there. The cardiac output, as we'd also expect, also goes down. So cardiac output is really this absolute measure of how much blood is pumped per unit time. In this case, the units are milliliters per second. We can have liters per minute. There are all kinds of different units. But the point is that it's volume per unit time. And we can see also a decrease as the animal gets smaller. No surprise there. What may be not quite so obvious is that the heart rate does the opposite. So the heart rate of a cow is, somewhere around that of a human. And as we move from the cow to the rat, we see the heart rate increases quite dramatically. So it really is, it's like a 5-fold increase in heart rate as we go from the big animal to the small animal. And that's an interesting phenomenon that we really ultimately should be able to explain if we really were to dive into this. And of course, the other piece, which shouldn't be a surprise, is the stroke volume. So that's the amount of blood that leaves the heart with a single beat. That's related to the cardiac output. And that, of course, goes down, just like it goes down in the overall cardiac output, but it goes down more dramatically. So the numbers on the right there are ratios of the first column to the last column. And so while the cardiac output went down by a ratio of 560, which is also a big number, the stroke volume rate, or the stroke volume number, went down even more dramatically, 2,700. And the point is, though, that these increasing heart rates and decreasing stroke volumes are actually coupled together to make the cardiac output, because the cardiac output is the product of the stroke volume, which is the amount of blood per one heartbeat, and the heart rate, the number of times the heart beats per unit time, and usually in terms of minutes. And so that product is what ultimately gives us cardiac output. But you see, it's not a symmetric kind of story. And there's some interesting explanations that might happen to be relevant there if we were to dig a little deeper. The last row of this table is the velocity in the ascending aorta. This is kind of an interesting idea of how quickly does the blood leave the heart as a velocity. So we know the volume is much higher in these larger animals, but what about the velocity, the speed at which it leaves? And it turns out that number, we don't have good numbers for cows, or I couldn't find them, but if we go through humans down to rats, we see the numbers are really pretty stable. I mean, there's some variability, but it's not dramatic, actually, compared to how dramatically different the stroke volume and different the cardiac output are. So it's always useful to think again in terms of these numbers and realize that at some point we have to kind of reconcile our understanding with these macroscopic observations that arise across different animal species. One of my favorite animals is the hummingbird. Now, you live in Utah, you've probably seen hummingbirds, and if not, just put a hummingbird feeder out, and eventually you'll have them sitting outside your window or on your patio, or whatever you can get to. And there are some beautiful renderings of the flight of hummingbirds, and we don't get to talk about that. That's another type of biomechanics. Really, really interesting. But the point is that the hummingbird has an amazing cardiovascular system. Not only is its flight so dramatic, but the cardiovascular system that supports it. And this shouldn't be a surprise. When you watch, you see here these hummingbirds hovering, moving around. You see the incredible speed of their wings. It's not hard to imagine that there's an enormous load on their cardiovascular system to keep those wings beating at this high rate. And so at some level we have to, I think, or at least might want to pay some attention and have some interest in this. Because it turns out that the heart is 20% of the body volume in a hummingbird, and that's enormous. So normally we're at a couple of percent of our body mass is actually our hearts. So you have a 10 times larger heart size by volume in a hummingbird than we do in a human. And hence this idea that there's got to be an enormous load, an enormous need for dramatically increased cardiovascular capacity in something that's moving, hovering, constantly zipping around. I mean, if you watch them, they do not sit still for very long. The heart rate also varies dramatically. We know in humans, as I said, the resting heart rate is somewhere around 60. Athletes get lower than that, of course. They're kind of famous for getting down into the 40s. The maximum heart rate of one of those athletes might be 200. I mean, the ballpark is 220 as kind of a nominal maximal heart rate. So that's, it's a reasonable range. I mean, it's not trivial to think about going from a resting heart rate of 50 to 200, just approximately. That's a fourfold increase. However, in hummingbirds, it goes from 30, which is already lower than our range, up to 500 when the hummingbird is just perching, not really flying, and then 1,200 beats per minute during high-speed chases. And if you've watched hummingbirds, they do a lot of high-speed chasing. So the range of heart rate here is dramatic, from 30 to 1,200. And how can that possibly be, right? There's got to be a physiological explanation for this. The metabolic side, again, it's not strictly cardiovascular, but just out of interest sake here, the mass-specific metabolic rate of a hummingbird is the greatest of all the vertebrate. And it's 100 times greater than an elephant, for example, to put it in some kind of context. So it has enormous metabolic demand. And as a result, has to be eating a lot. It consumes constantly. It's eating, as you see, it's constantly sucking nectar, trying to find energy to provide that enormous metabolic demand. So it eats half to 8 times its body weight per day in food, depending on the food source, which is pretty amazing. We wouldn't do so well with that kind of amount of eating. The respiratory rate also, we'll talk about respiration later, also exceedingly high, 250 breaths per minute at rest. So again, much higher than the 14 or 15 that we nominally have at rest. And then, of course, there's this mechanical part of it's flying and it's diving and it's maneuvering, which is so dramatic to watch. And the dives that hummingbirds create can achieve accelerations that are 7 times or even 10 times as high as the gravitational attraction on the Earth. So it's called a 7 to 10 G acceleration, and reach a speed of 100 kilometers per hour. So it's 60 miles an hour. So these are amazing creatures, and just one example of the kind of physiological challenges and yet interesting domains that we can see in different animal species. All right, so let's go back to some more generic stuff. So the cardiac cycle is something we've seen before in the electrical context. Now we have to see it in the mechanical context. My favorite diagram for all this stuff is called the Wiggers diagram. Wiggers, of course, was a physiologist who developed this diagram. It's a really rich source of information, and we'll see it again and again. We'll discuss it in the discussion sessions and whatever else. But we should create a mapping in our brains between, for example, what we see here of the mechanical steps. These are 5 steps through the cycle of a heartbeat in the mechanical sense, and then the equivalent behaviors that we would see across a heartbeat that's revealed in the Wiggers diagram. And the Wiggers diagram typically are organized like this, where you have left heart, right heart, you have functions of time, different functions, pressures, volumes, flows, all these sorts of things. So it's a really rich snapshot of the behavior of the heart from a mechanical perspective. And oftentimes we throw in an ECG to provide kind of a temporal reference, because ECG is what, as we've seen, we measure that easily in the lab. It's much easier to measure an ECG than any of these other variables. So where they occur in time is important. And again, you'll see, as we saw in this cellular domain, for example, that the QRS, which we all know now corresponds to the electrical spread of activation through the ventricles, precedes the peak of the pressure in those same chambers. So again, that's another one of these mechanisms. Think about it a little bit, go back to the cellular picture. If it's not clear, go back and look at that video again. When we have the upstroke of an action potential, there's a delay between that time and the time of maximum contraction of that cell. That same, the effect of that delay is we see a similar effect here at the whole body level, where the ECG electrical activation or excitation precedes the mechanical response. So we have electrical systole coming before mechanical systole, and that will be important to keep in mind as we study different aspects here. One thing we can do from the Wiggers diagram is extract information from it and plot these variables against each other. And there's one particular form that's quite useful you may have seen before. We can take the pressure curves and the volume curves, which are functions of time in the Wiggers diagram, and plot one against the other. So it's a little bit like in the ECG lab, where we had ECG leads against time, and then we plotted them against each other in order to create vector loops for the vectorcardiogram. Same mathematics here, if you will. It's not deep math, but conceptually interesting is to take these 2 parameters and plot them. And again, we get a loop, just like we got loops in our vectorcardiogram. We have loops here, and because we're taking pressure and volume, we call these pressure volume loops, or PV loops. Now, this is a great snapshot, if you will, of the mechanical behavior of the heart. This is why these PV loops are important. There are actually devices now available to generate these PV loops in patients, in people. They involve a catheter. They're not lightweight to do, but it's possible to make these measurements. Pressure, you can imagine, is a pretty easy measurement to make. A simple catheter will do that. Volume is a lot harder. But let's say we have the pressure volume loop. We can see the entire heartbeat played out in that pressure volume loop. We can walk around that loop and associate each point in time on that loop to a phase of the heart cycle. And we should become somewhat comfortable. We should be able to label the major events in that cardiac cycle on that PV loop. So for example, those 5 stages you saw in the previous diagram, we should be able to map those onto this loop. We should be able to map on when the valves open and close, and when contraction occurs, when different things occur. We should be able to put those on this and then capture a sort of nice full picture of the pressure-volume relationships and what they tell us about the function of the heart. Because there are some simple metrics that we can extract from this. One is the amount of blood that leaves the heart with every heartbeat, or the stroke volume. And that's simply this horizontal distance between the right side of the PV loop and the left side of the PV loop, right? That's the amount of blood that leaves the heart. That's the shortening. And we can also measure things like the maximum pressure differences that we would see. That's the vertical distance. We can extract lots of key values from this PV loop. In fact, at a highest level of all, if we integrate that loop, which means the same, we get the area inside that curve, that's actually a direct metric of the amount of work that the heart achieves. So when a heart starts to fail, when it starts to do less work, then this pressure volume curve will get smaller. Or when we exercise, we can anticipate this pressure volume curve getting larger. So it becomes a really useful signature of overall cardiac mechanical behavior. And that's why we have to become pretty comfortable actually with how we use it and how we imagine its utility. And again, it's a visualization that's very rich, and we should be comfortable with it. Now here's just one example of what I mean. What happens when we mix that concept of preload or pretension that we talked about in the cellular and the tissue domain into the space of the whole heart, where we call it the Starling's law, or the Frank-Starling response of the heart. Law is a little strong here. There's some argument about how big an effect Frank-Starling is, but I'll leave that controversy. But the point is that Frank-Starling does seem to have some effect, and here's how we capture that effect in a pressure volume curve. So in the middle, the sort of thick dashed line, you see the pressure volume loop for a control situation. And if the preload goes down, which means there's less venous return, right? Less blood comes back from the right side of the heart, does not fill the ventricles as much, does not generate as much pressure in the ventricle, then the response is a smaller pressure volume loop and a smaller stroke volume, right? Because the width of that pressure volume loop will actually get smaller. The whole loop will move to the left, but it'll also get narrower. You can convince yourself if you make a measurement on this. That's what happens when we have a reduced return, a reduced preload, a reduced pretension. Of course, the other opposite is we get an increased pretension. Exercise starts, lots of blood gets removed from the venous side, pumps into the heart, has to then be pumped into the systemic and pulmonary circulations. That ultimately leads to an increase in the PV loop. And you see that increase happens left to right, so pressure volume changes. You also see the curve gets taller, so the pressure, the peak pressure that we will see will go up. And that's actually a phenomenon that we'll investigate both in the lab and in the lecture series. So we see that changing conditions on the heart can be measured very nicely in an integrated sort of way in these PV loops. So that's really why they're relevant. And as physiologists, especially quantitative physiologists, this should make us happy. We should enjoy this capability and try and make use of it. So the Frank-Starling mechanism is one of those intrinsic regulation systems that we talked about in the context of the cell and the tissue. Just as we have in the cell and the tissue, we have extrinsic means to change the PV loop, to change the performance of the heart at any given time. And of course, you should already be thinking the key factor here will be calcium. Calcium at the cellular level changes the contraction that propagates all the way to the whole heart level. And we should also be thinking very quickly of the autonomic nervous system as the main extrinsic means of regulating the heart, and that's certainly the case. And as we know, the role of the autonomics are to balance two different types of forces in order that our bodies stay at a homeostatic or relatively constant level, or at least at some set point. Remember this whole negative feedback story? We have a set point, we have negative feedback, and the whole goal of that system is to keep our system at that set point. We'll talk later about what that set point means in the context of the cardiovascular system. But at this point, we can sort of anticipate that there's going to have to be a system in place in our bodies that can either upregulate the heart function or downregulate it, depending on whether we're exercising or sitting around watching TV. So, and indeed there is, and the mechanism by which that happens is calcium. So we alter calcium control, our calcium levels in the heart, just as we described in the previous video. Now, what's the effect on the PV loop? Same sort of idea. We get an increase in contraction that will make a wider loop, PV loop, more stroke volume, more cardiac output, higher pressures. Again, the higher pressure will increase our cardiac output. And then if we could see that, and that's the downside of a PV loop, if we could see the rate, because it's not we can't read that directly from a PV loop, but if we watch how fast that loop traces, we can even get heart rate. And out of the heart rate and the stroke volume, we can determine the change in the cardiac output. And so the question that comes up naturally is, what is the balance between these two systems, the intrinsic and the extrinsic? And we'll get to some of that even a little bit later, but the key point to keep in mind here is that there has to be some means by which the extrinsic can work through the brain, the nervous system, and also, in the case, let's say, of a transplanted heart, how it can be, or whether it can be regulated. Now, the intrinsic system will still work in the context of a transplanted heart. This is a heart with no nervous system attached to it. That will certainly still function as we would expect. But what's the mechanism for the extrinsic control? And of course, that's the endocrine system. It's the amount of these compounds, the same neurotransmitters that we have in the nervous system as part of the autonomics, that are secreted into the blood and systemically circulate and activate the heart, cause it either to upregulate or downregulate, depending on the nature of it. But at the end of the day, Frank-Starling still works. That's still an intrinsic means, but the extrinsic functions change the curves. They shift the curves up, for example, if it's if it's a positive inotropic effect, and decrease it on the other hand. So the effect of that is just a propagation of what we described at the cellular level. We have the same neurotransmitters, epinephrine, norepinephrine, and their effect on the whole heart will cause a change in the PV loop, a change in all the serious functions, but will just be an upregulation of what we've seen before. And even these contraction curves that we talked about in the context of tissues, they also just get shifted. The behavior is still the same, they just get shifted. Now finally, I'm going to include or point you to a couple of videos that you can watch that describe contraction of the heart. And they describe contraction of the heart in normal condition. They also talk a little bit about heart failure. And so they're hopefully useful to fill in or summarize some of what I've told you. But the point is that the overall behavior of the heart is really just a propagation of the cellular behaviors. And then finally, we talk a little bit about modeling. You've played with the model of the membrane. We're also going to play with the model of the whole cardiovascular system eventually. We're not quite there yet. But we also have models that will simulate the mechanical behavior of the heart. And so here you see a video. This is from a piece of software that came from the INRIA lab. So these are the sort of national lab system in France. And these are quite dated now, but you get the basic idea that these are three different views, two slices and an exterior view of the heart contracting. Color here is showing the amount of tension developed. And they give you a sense of, again, the models are better today for sure, but it's definitely possible to write down equations, to come up with numerical expressions for those equations, to simulate those on large computers, and ultimately recreate mechanical activity. And this is a very hot topic of research right now. A number of labs are moving into this domain. Labs that have extensive experience in electrophysiology are now moving into this domain of cardiac biomechanics. Of course, because at the clinical level, heart failure is becoming a very, actually rapidly increasing concern among physicians, among cardiologists. So this is both the motivation, and with bigger computers, we can achieve a lot now with simulation. So again, big topic. Don't have time to get into it seriously, but it's definitely a domain that you might be interested in pursuing in your own research areas, whatever those happen to be. Finally, a little bit about animal hearts. I can't resist it. I'm sort of fascinated not just with hummingbirds, but with other things. There are lots of different cardiovascular systems in different animals. So we've, I think, talked a little bit in the past about systems like the mollusks, which don't really have a closed cardiac system. They have a system that sends blood out that eventually leaks across or diffuses across from what we would call the arterial side to the venous side. We've seen that before. There's another body or type of animal that has a rigid pericardium and uses complicated pressure differences to create contraction and create pumping in this heart. So instead of an elastic heart like we have, with an elastic pericardium, like we or the cows or most mammals, there are animals that have these rigid pericardium. Fish, of course, big category of critters that we care about more or less, and they, of course, are breathing differently. We'll talk more about them in the context of respiration. But they also have a different cardiovascular system, and you can see here schematically that the blood goes through the tissues just like ours does, gets deoxygenated just like ours does, but there's a secondary circulation that it goes through. There's a single ventricle rather than two ventricles, and the blood goes through the lungs and then continues from the lungs directly into the rest of the circulation. So there's not a second pump that pumps the blood, the oxygenated blood, into the rest of the body. And why would there have to be? If you only need one pump, maybe that's a evolutionary advantage. And so fish definitely have different systems. They're water breathing, or fish that are capable of extracting oxygen from water. There are also fish that can breathe above water, and so they have a dual system. They have gills and an air breathing system, and so they end up with this kind of crazy complicated circulatory system like you see here. Again, a single chamber heart, just like we saw with the other fish, but a lot more complexity in the actual cardiovascular system. The last one I want to talk about is, well, there's two I still want to talk about. The one that's probably most relevant to us is the frog, because we're going to do a frog study. And as we've hopefully seen and remember, the frog has one ventricle. So how can that possibly work? That's a question we'll discuss more in the context of the lab. Here you see an image, schematic of course, of the blood flow through the frog heart, and you see very clearly there's a single ventricle. Here's another, slightly more schematic version that describes some of the additional details. But the point is here that a heart can have a single ventricle and still provide separate circulation for the pulmonary and the systemic. So there's a lot of range here. And reptiles are sort of between amphibians and us in the sense that they have a two ventricle heart, but with a opening between the two ventricles. So they have, if a human were born with this condition, you'd say they had a defective septum, or a septal defect, in this case a ventricular septal defect. And, but again, there are ways that we can explain this, talk about this, and this will work mechanically. Obviously reptiles that are not crocodiles, function this way and do just great. Now finally, I want to talk a little bit about the fetal heart. It's not an animal heart. Well, I guess we're animals too, even as humans, but this is an interesting case in which the heart changes its function quite suddenly when you think about the dramatic change in condition from before birth to after birth of the fetus. So the heart has to function in a very different system. So obviously when the baby's in utero, there is no functioning lung system of its own. It has lungs, but they don't function. It has no access to air. So the oxygen has to come from the fetal blood or from the placental blood. And indeed, that's what happens. You can see that in this diagram here, that these numbers you see are percentages of the blood flow. And so 70% of the blood flowing into this heart comes from the mom, essentially, and into the heart. And there's some that's flowing back from the rest of the body that also comes in through the superior vena cava especially. So we have blood that's oxygenated already mixed with blood that's not oxygenated, and somehow that has to get out the other side of the heart without having lungs to oxygenate or additionally oxygenate that blood. And there are two important functional and anatomical differences. Again, structure function. If we observe, there's a septum between the two left and right atria, of course, but there's an opening in the fetal heart called the foramen ovale, and that opening, that gap or break in the septum, provides a direct blood pathway from the returning blood, which again is oxygenated in this case, from the right atrium into the left atrium, then through the valve into the left ventricle to be pumped to the rest of the heart. There's another connection that's unusual, and that goes from the pulmonary artery. So you see the pulmonary artery does leave the right ventricle, just like a normal heart, and that blood flows up and does go to the lungs. I mean, the plumbing is there to go to the lungs, but only a very small percentage actually goes to the lungs and doesn't really achieve anything of utility. So in this case, something like 7% goes to the lungs, and the rest of that part of the 67% that flows, leaves the right side of the heart through that opening, goes through another unusual opening called the ductus arteriosus that connects directly the pulmonary artery with the aortic arch. And that way, this partially oxygenated blood leaves from the right ventricle and goes directly into the systemic circulation. And of course, these two openings have to close directly after birth in order for normal respiratory function to pick up and to provide the oxygen needed from outside air and not from the mom, who is no longer connected, of course. So the bottom line here is that, hearts have many different structures, and different animals have different structures. And as physiologists, we at some point may have to entertain any of them if we're working with an animal model or looking for an artificial organ. So it just helps us to appreciate that there's a huge degree of richness and diversity in the solutions that different animals have found to this general problem we all share of moving nutrients, oxygen, byproducts, toxic substances, whatever, moving those around through our bodies. And that also leads to a similar variety in the structure and function of the hearts themselves, especially in their mechanical and their biomechanical aspects. So that's, again, sort of as much time as we have to get into this whole topic. Hope that was at least interesting, of course, but hopefully gave you a good sense of how we are able to couple from the electrical behavior to the mechanical and describe in at least some high-level terms what the mechanical parameters are and what the mechanical functions are that we always have to be aware of when we're working with animals or working with patients who have either heart diseases and where the vascular cardiac system plays a role. So, hope that was interesting. Thanks again for your attention, and we'll see you soon.